Hi everyone, welcome back to my channel. In this video, I'm going to talk about the concept sickle cell anemia or sickle cell disease. Sickle cell disease is a blood disease in which the individual will be having defective hemoglobin of RBC. Because of this defective hemoglobin, the RBC looks like sickle shaped. So this individual will be having sickle shaped RBC. Normally, the normal hemoglobin is represented by HbA. And this HbA will be having, or hemoglobin will be having, 4 polypeptide chain. Out of 4 polypeptide chain, 2 are termed as alpha and other 2 are termed as beta. Each of this alpha chain will be having 141 amino acid residue, whereas each beta chain will be having 146 amino acid residue. So that is a normal hemoglobin. The hemoglobin of sickle cell anemia patient is represented by HbS. Again, this HbS also have 4 polypeptide chain. Out of 4 polypeptide chain, 2 are represented as alpha and other 2 as beta. The 2 alpha chain will be normal. Normal means it will be having 141 amino acids like that of the normal hemoglobin. Whereas there is a difference in the beta chain in the sickle cell anemia patient. Difference means there is a difference in the 6th amino acid. Each beta chain at the 6th position, it will be having the amino acid named valine instead of glutamic acid. In the normal individual, each beta chain at the 6th position will be having the amino acid called glutamic acid. Whereas in the case of sickle cell anemia patient, at the 6th position of the beta chain, it will be having valine instead of a glutamic acid. So this change in the amino acid sequence from glutamic acid to valine at the 6th position of each beta chain is mainly due to single base change or substitution from CTC to CAC. As you can see in this diagram, this is a sequence in the case of normal hemoglobin. This is in the case of a sickle cell anemia patient. In the normal hemoglobin, at the 6th position, it will be having the codon CTC. Whereas in the case of sickle cell anemia, it will be having CAC. So, T is replaced by A here, or single base T is substituted by the base A here. So, types of mutation is single base substitution. Because of this single base substitution from T to A, during protein synthesis, the mRNA will be having 2 different types of complementary codon in 2 different cases. In the first cases, the mRNA will be having complementary codon, that is GAG. So here C pairs with the G, T pairs with the A, C pairs with the G. So this mRNA will be having the complementary codon GAG in the normal case. Whereas in the case of sickle cell anemia, the mRNA will be having the complementary codon GUG. So here C always pairs with the G, A pairs with the U here, not T, because it is RNA. So in RNA, T is not present. Instead of that, uracil will be present. So A pairs with the U and C pairs with the G. So here the mRNA will be having the complementary codon GUG. So from GAG it changes to GUG. So in the first case, or in the normal case, GAG codes for glutamic acid. Whereas in the second case, or in the sickle cell anemia case, GUG codes for valine. So that is how at the 6th position of the beta chain, the glutamic acid is replaced by valine. So it is mainly due to the single base substitution. So because of single base substitution, the valine is present at the 6th position of the beta chain instead of glutamic acid. This mutant hemoglobin now undergo polymerization. Under low oxygen tension, there is a change in the structure of RBC from normal biconcave disc shape to elongated sickle shaped RBC. This sickle shaped RBC or sickled RBC will be having short lifespan of about only 20 days, and they carry very less amount of oxygen, or oxygen carrying capacity of this sickled RBC is reduced, due to which the patient suffers from anemia. As I already told, the gene responsible for sickle cell anemia is denoted by HbS, and this sickle cell anemia is a autosome linked recessive trait. Autosome linked means this abnormal gene or gene responsible for sickle cell anemia is present on the autosome. In particular, it is present on the chromosome number 11. So it is an autosome linked recessive trait. Recessive trait means only in the recessive condition the symptoms of sickle cell anemia appears in that particular individual. Or in other words, when both the chromosome of the homologous pair, if it has this abnormal gene or gene for sickle cell anemia, only in that condition that particular individual will suffer from the symptoms of sickle cell anemia. So these are the three conditions in which this abnormal gene or gene for sickle cell anemia can be present in different individuals. In the first condition, that is HbS, HbS means both the chromosome of the homologous pair will be having this abnormal gene or gene for sickle cell anemia, or it is a recessive condition. In this condition, that individual will suffer from sickle cell anemia. So in this condition, that individual will die in the early stages of his life, because this individual will suffer from lethal anemia or severe anemia, because of which that individual will die in the early stages of his life. So he won't reach a sexual maturity. So whenever we talk about the sickle cell anemia patient, we are talking about the carrier. Carrier means in this out of two homologous pair, one of the chromosome will be having the normal gene or the dominant gene, whereas another pair will be having this abnormal gene or gene for sickle cell anemia. So this individual will be normal. Phenotypically that individual will be normal, or he or she does not show any symptoms of a sickle cell anemia, but they are the carrier. Carrier means they carry the gene for this sickle cell anemia for the next generation. So they carry that particular trait, that is sickle cell anemia trait, for the next generation. The third condition is HbA. HbA means it is normal condition. So both the chromosome of the homologous pair will be having the normal gene. So no abnormal gene is present, because of which that individual will be normal. Now we'll discuss about the mode of inheritance of sickle cell anemia in different conditions. There are mainly two conditions exist. In the first condition, the carrier male marries a carrier female. So in this condition, both the parents are carrier for sickle cell anemia. So in the first condition, the carrier male can produce 50% of the gametes having a normal gene and 50% of the gametes having this abnormal gene or gene for sickle cell anemia. Similarly, this carrier female can produce 50% of the gametes having the normal gene and 50% of the gametes with the abnormal gene or gene for sickle cell anemia. So when this gametes having normal gene combines with this gametes having normal gene, it can produce normal individual. HbA. HbA means normal individual. Similarly, when this HbA combines with HbS, HbA, HbS, so it can produce carrier. Again, this HbS gamete, when it combines with HbA, HbS, HbA means it produces carrier. Similarly, HbS, when it combines with the HbS, it produces sickle cell anemia individual. So HbS, HbS means sickle cell anemia individual. So in the produced offspring, 25% of the offspring will be normal, 50% of the offspring will be carrier, and 25% of the offspring will be sickle cell anemia. So this sickle cell anemia individual will die in their early stages of life because of lethal anemia or severe anemia. Whereas this carrier will be normal, but this carrier will carry that abnormal gene for the next generation. So this individual will be phenotypically normal, but they produces 50% of the normal hemoglobin and 50% of the abnormal hemoglobin. Okay, so that is in the first condition. In the second condition, where the normal individual marry the carrier. Carrier means having HbA, HbS. In that condition, so this normal male can produce all the gametes having normal gene, whereas this carrier woman can produce 50% of the gametes having the normal gene and 50% of the gametes having this abnormal gene. In this condition, when this normal gametes having normal gene combines with this gamete having normal gene HbA, HbA, it can produce normal individual. When this HbA combines with HbS, it will produce carrier. So 50% of the offspring produced will be normal and 50% of the offspring produced will be carrier. So carrier means they produces 50% of the normal hemoglobin, 50% of the abnormal hemoglobin, due to which they are phenotypically normal, but they carry that abnormal gene to the next generation. As you can see, we didn't talk about the sex of the offspring produced here, because it is a autosome linked trait. Autosome linked means the gene responsible for the sickle cell anemia is present on the autosomes, not on the sex chromosome. In the autosome linked trait, possibility of both male and female being affected is safe. It does not depend on the sex of the individual. Again, even we didn't talk about the recessive condition here. The parents what we have taken in both the conditions are either carrier or normal. But we didn't took this recessive condition because this recessive individual or individual with the sickle cell anemia will die in the early stages of life. They won't reach the sexual maturity. They will die before reaching the sexual maturity. That's why we didn't took this individual as a parent. So the possibility is either being the carrier or the normal individual. So that is all about the sickle cell anemia and its inheritance. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.